Welcome to A Window on SAHMRI, where we take you inside South Australia's independent not-for-profit health and medical research institute. Each episode, we get to know the people driving our life-changing research, getting into what motivates them personally and how their work is delivering a brighter, healthier future for all.
Chris, why is cardiovascular disease the leading cause of death in Australia and worldwide?
Yeah, so it's really a result of having predominantly too high cholesterol levels in the blood. And when you have that, it causes the build-up of fatty deposits in the blood vessels of the heart. And when these deposits become too large, they will completely block off the blood supply, and therefore not enough oxygen and nutrients get delivered to the tissues of the heart, and that's what causes a heart attack. So a lot of people have high cholesterol. It increases naturally in most people with age. A lot of people also, just through hereditary reasons, they will have elevated cholesterol levels, and there's a lot of reasons biologically why that can happen, and that's the key cause, and that's why.
Why does cholesterol typically go up as we age?
I think it's to do with our liver function, so that is not as efficient at clearing cholesterol from our bloodstream and then sort of removing it. So it just starts to accumulate gradually over time.
How much is lifestyle a part of this picture?
Yeah, I mean, lifestyle is a big part of having high cholesterol levels. Obviously, diet, high cholesterol diet, saturated fats as well in meat, they all increase cholesterol levels. If you don't exercise, that also contributes to high cholesterol. So if you do exercise regularly, it can actually impact on your cholesterol and reduce it by up to 20%, which is pretty significant. So lifestyle does play a big role, but it's not enough. So even really fit and healthy people with no obvious risk factors will have heart attacks without expecting it. So there's a lot more that researchers need to do to understand why people have elevated cholesterol levels and how we're going to reduce it.
What can be done about that at this stage with research, healthy people who still have these problems or people who aren't so healthy? What can we do to help mitigate and prevent this from happening?
Yeah, so with the healthy one, that is really the holy grail, because at the moment, people just do this standard kind of risk factor assessment and they're just not picked up. So that is something that researchers need to do. We need to get better at, maybe there's a standard blood test that everyone has at a certain age that can detect the presence of even the earliest stages of the disease or some kind of susceptibility to developing it. But nothing like that exists at the moment. Some of my research does try to address that, but that really still remains the holy grail, because if you can prevent heart disease early, or identify it early, it is a lot more treatable with drugs and reversible. So that is really important. Now, for people that are less healthy, there are drugs, cholesterol-lowering drugs. They're really common, actually. Many people are on them, a lot of males, and also females from the age of 45 upwards. They're on the statin drugs. They work by inhibiting cholesterol synthesis, and they can really bring people's cholesterol levels right down to a really acceptable rate, which is about 4 millimoles per litre.
So healthy people whose risk doesn't show up on a standard test, is there anything that they have in common with each other that does show up that perhaps isn't on that test? Or
that's a difficult question.
Am I asking questions that don't have answers?
So no, I think in this case that is still something we don't know. So, but there are definitely people doing it. So we believe the answer is somewhere in sort of the omics technologies. So people will take the blood vessels or the blood samples from people that are healthy versus those that have disease, and then they'll try and pick up or those that after doing the risk factor assessment are apparently healthy but will go on to have disease. And then we're trying to understand what proteins, what genes, what other kind of like microbiome that they might have in common, so we can pick up those people that would be assessed as being healthy and very low risk that might go on to have the disease. So Just to reiterate, I think the answer lies in some of these omics technologies where we're doing really broad range screening of all the possible genes that might be regulating it or proteins. And the work that we've got a novel omics platform which is called glycomics, which basically looks at all the range of the different sugar molecules that are in our blood, and they can have a lot of regulatory actions in cholesterol metabolism as well.
But especially tricky because you don't exactly know what you're looking for.
Exactly. So it's going to take really well phenotyped individuals, large clinical trials, and a lot of this sort of biobanking of samples from people across and from different demographics as well, in order to try and do this. Yeah,
it's a mystery.
It's a black box.
But one that you're happy to work on.
Yeah, so we're working on a small part of that with a team. It was an MRFF government-funded project. Myself and Professor Peter Soltis, a cardiologist, are running a trial where we're taking really a beautiful snapshot of people's heart using this amazing sort of photon counting tomography device that they have at SAHMRI, and then we're characterising the range of sugar proteins in the blood by collaborating with the leaders in the field that can measure those things.
You published a paper in 2022 which found that 90% of researchers in cardiovascular research have considered leaving research. So it seems like a crazy stat. What's behind that?
Yeah, I mean, it isn't just specific to cardiovascular, but for whatever reason, we might be a little bit more susceptible to losing some of our amazing up-and-coming scientists, and it's a funding problem. So you come out of your PhD, there's a limited time to try and get a fellowship to support your own salary, and then to try and get money to run your research. And they're highly competitive, these fellowships. And I think people that have done their PhD, they see that and how hard it is, and they either immediately just get turned away from that and go to industry or some other career, or they'll test it out for a while but then eventually get burnt. And I think only maybe 10% of people will make it through to staying in science, and even less of those will go on to become independent researchers and can consistently be successful at running their own research team.
So you must have come across a lot of promising young scientists in your time who didn't end up making it because of that reason.
Yeah, a lot of people, unfortunately. So I've had a lot of really great postdocs that they decide to move to medicine because it's obviously a little bit more obvious that you'll have a job, and a lot go to industry as well. And it's a real shame. Some of the brightest minds will lose, and they won't be there to discover new things. Yeah.
Seems like we should be spending more funding on the number one killer in the whole world, doesn't it?
Oh, absolutely. And I am a real advocate for that, even though, that's right, heart disease is the biggest killer. We actually only get about 10% of the research funding that cancer do. And I think we shouldn't change that. I think cancer deserves to have all of that research funding, but I think heart disease should at least be equal to that. And for whatever reason, it isn't as well supported as some of the other diseases. And I do believe it's because there's a perception that heart disease can be solved by just lifestyle choices, but it's a lot more than that. That can explain about 20% of heart disease. The rest is still this black box I talked about.
Yes, perception is everything though, isn't it? And I suppose that's another challenge with heart disease is the fact that it's so gradual often. Someone might have some of those symptoms and be experiencing high cholesterol and living with varying degrees of heart disease, but it might go on for decades before they actually have a heart attack.
That's right.
So often not quite as sudden until that moment comes or in your face as something like particular cancers.
Yeah, I think that is also part of it. So you can go on without and be building up these fatty blockages in the blood vessels of your heart for decades without any symptoms, experiencing any symptoms, except maybe a shortness of breath at some point and not think much of it.
And that can lure people into thinking it's not that big of a deal because the body can put up with a lot.
I think so. But then what will happen is you undergo these fatty blockages will completely block off the blood vessel, or sometimes these fatty blockages become what we call unstable, and they undergo this rupture event, which is an explosion, and then we get a full blood clot that completely also blocks off the vessels. And they're the ones where all too often it results in sudden death.
It's a bit of a ticking time bomb, but it can be a ticking time bomb that extends over years and years and years. So
exactly,
forget that it's a threat.
Yes.
What drew you to heart research?
Yeah, so my honours year was on lipid metabolism, and I did really enjoy just understanding how the liver regulated cholesterol and the proteins and the biology around that. And then I did that also in my PhD. And then I was lucky enough after my PhD to get a postdoctoral position at Oxford University. And in that lab, I learnt about atherosclerosis. It was in the early 2000s at the time. And at that time, they just invented sort of a mouse model of these fatty blockages in the heart, which is called atherosclerosis. So we had this first mouse model that mimicked relatively well the same sort of heart disease-like scenarios that we get in humans. And so I worked on that, and I used novel these gene transfer strategies to block inflammation, which was also sort of new technology at the time. And I just really loved it. I really found it very interesting and felt that there was a real need there to try and reduce, this biggest killer that we have in the Western world.
So you came in at a time when you felt like you were on the cutting edge.
Yeah, and that was also a reflection of how great the lab I was in. I was really lucky, and I had all of sort of the latest technologies and some of the smartest minds in the field working with me at the time. And so I think that's also why I really enjoyed it. But I think, and also because my project was on this fatty blockages, but also the inflammation around that. And it was the first time that inflammation was being associated with how heart disease starts to develop. So I did really feel like we're sort of on the cutting edge of all the new information that was coming out. So
there were a lot of firsts.
Yeah, there were a lot of firsts. Yeah, that's right. So the publications, I got really very nice high-level papers that came out in succession in my 5.5-year postdoctoral time, because I think we're always doing something really novel and new. Yeah.
And did you have mentors that made a big impact on your career?
Yeah, I did. So my supervisor in Oxford was Professor Keith Channon. He's an interventional cardiologist, but a very, very brilliant scientist as well. And just the way he would just have amazing ideas, but just know how to solve problems and know the best way forward. He really did help me and support me, but push me as well. And I still today will be trying to manage someone in my team or work out a problem and think, what would Keith do? So yeah, he's definitely, and he's still always there for me. If I need a support letter for something or whatever, he's still there for me. So it's a big one.
Special connection. You try to emulate that with the young people that you supervise?
I try to, because he did really inspire me, and always try and do a really good job at everything. And just, I think his problem-solving ability was really the most inspiring. You'd go in thinking, there's no way I'm going to ever get this to work, and you'd leave a meeting with a spring in your step. And I do try and pass that kind of enthusiasm for discovery and science on to my team at the moment, and I hope I can keep them in science for as long as possible.
So there's always a way.
Always a way. Yeah, there is always a way. I really think so. Never like to give up. Yeah.
You went to Sydney and then came back to Adelaide.
That's right.
Why did you come back to Adelaide?
There was a few factors. I had a baby, so I had my son Henry, and we were living in me and my husband were living over in Sydney with no grandparent support, and all our family and extended family live in Adelaide. So that was a big factor for moving back to Adelaide. Another thing at the time was that Steve Nichols was at SAHMRI, and so obviously a really great cardiovascular person to work with. So he was a big part of recruiting me back to SAHMRI. And I was also lucky enough to receive the Lynn Huddleston Heart Foundation Fellowship, which is a South Australian-based fellowship. So I got a 5-year career package thing, yeah.
I'm sure luck had nothing to do with it.
No, I was very pleased to well, they only gave out one, so always a bit of luck then, yeah. Yeah.
You're rather humble, Chris. So what's kept you at SAHMRI and kept you in heart research, given that it is such a difficult field? Yeah,
well, look, I really like the biology of this atherosclerosis process, these fatty blockages. I'm really interested. I'm really looking always for new genes, targets, proteins, so we can try and reduce it. So I do really like that. I think we've got some really cool models that we work with as well. But I really like SAHMRI as well. I think it's got a really good culture. I think the leadership is amazing. It always has been. And I like all the equipment. I mean, it's a really great lab space with that's really well supported. It's my favourite institute that I've ever been at, yeah.
Tell us about some of your proudest achievements to date.
Yeah, so I think some of my latest work I really like. I've been, lucky enough to work with, I guess, fundamental scientists. So they'll be nanoparticle experts or photonics experts or chemists. And by bringing in these kind of multidisciplinary fields means that my science, I believe, has become more innovative. And one of these is a collaboration with a nanoparticle expert called Gang Zhen in Toronto. And he developed these tiny little spherical particles we call nanoparticles. They're only about 50 nanometers in diameter, which is 3,000ths the size of a human hair. So they're very small. And
how did you measure that?
I calculated it in my head. That's why it took me so long to get it out. But so a human hair size is 3,000 times bigger than these little nanoparticles. Wow. But they're spherical in shape, and he had previously invented them for the application of cancer, but we've now worked them into our heart disease models, and they beautifully will light up these fatty blockages in the heart. So I think they're a really nice way for early disease detection. They can go in and they have the ability to emit a fluorescence light, which you can detect just by a fluorescence detector. But not only that, you can incorporate into the core of these little particles different therapies that can also act to reduce the size of the fatty blockages as well. So they're kind of multifunctional little things. And we've got a paper now nearly accepted in a good journal on all of this work that's been run by my postdoc, Victoria Nankervell. So I am really proud of that. She presented that at a conference last year called the Cardiac Society Australia New Zealand, and she won the Ralph Reeder Prize in this sort of young investigator competition, and it was the most prestigious competition for a young investigator in the cardiovascular field. So I am really proud of that, of her, obviously, and the data that's come out of that work, yeah.
So those fatty blockages, once you see them light up, is there something that can be done to actually target those specific areas?
Yes. So I guess it's two parts. So what we can do is that we can, when we load the different therapies into these particles, we can then use that lighting up to track if it's making them smaller or not. But you could use them in their own application in a way just for their ability to detect plaque. So ultimately, these little particles, we need to get them FDA or TGA approved so they can be infused into humans. But if that was the case, then we could detect, after a simple infusion, we could detect the earlier stages of these fatty blockages in the heart for people. And then we would know, okay, we've got to put these people immediately on cholesterol-lowering drugs. Maybe they need to have a stent put in or something like that. So interventions can start happening.
So another way of finding out what's going on inside the body, perhaps in even more detail, well, definitely.
Yeah.
And one of those things that we started talking about at the start, where it's another way of picking up something that perhaps would get missed by traditional methods.
Yeah, that's right. And what's amazing about these is they're highly fluorescent. It's that there are a few kind of what we call imaging agents that can detect plaque, but they're not as bright, or the resolution to see the earlier stages is not good enough. But this overcomes a lot of those problems by having these little particles go in. They're taken up by the key cells that are there in the earlier stages of fatty blockage development, and they really emit a very strong fluorescent light.
And in that article that you wrote a few years back, you noted that heart health research had hit a plateau in your view. Have we busted through that since then, or where do you feel like the field's at?
Yeah, actually, I would say there has been a real leap recently, in recent times. So I think that the newest breakthroughs are with the ability to use genetic material. So there's something called CRISPR, which basically acts like a pair of scissors to exactly snip out a gene that you want and remove that protein from that gene being made at all. And it does it to your DNA, and that means it's permanent. So people have developed this. I think people probably have heard of CRISPR, but it's basically a pair of scissors, and it's a bit of genetic material that gets packaged also into these little nanoparticles, and it means that with a single infusion of this, it will knock down these proteins forever for a person and have amazing results in lowering cholesterol. So that's what's been done in a number of trials recently. So they've used various versions of deleting a protein. It's called PCSK9, but when you delete it or remove this protein, it really lowers your blood cholesterol levels. And rather than having someone take statin therapy, cholesterol lowering therapy every day by a tablet, it means a single intravenous delivery might be enough for the rest of your life.
Oh.
And this is now in the just really taken off in the last year or two, and it's moving into human, and it is moved into human trials in pilots. They've advanced through and into phase 1, phase 2 clinical trials, and has been really impressively successful.
So we know that this protein is spurring cholesterol.
Yeah. So this protein, what it would do normally is it causes degradation of a good protein that would remove the cholesterol from your bloodstream. So by removing the bad protein, it keeps the good one there to do its job.
To work properly.
Exactly, to work properly. That's right. And so that particular bad protein, though, has been the only target at the moment, and cholesterol lowering is the only strategy that they're using to reduce heart disease. And so there is a real window for a bunch of other targets in the future as well that might have additive effects on top of it.
That's exciting stuff.
Yeah, it is, yeah.
And you work in discovery science.
Yeah.
Can you explain what that is?
Okay, so I love discovery science, so I can read things and go, okay, that might work in my models, or maybe if we delete this, or does this gene have something to do with new blood vessel formation, and we can just go and do it and literally find out for the first time the role of something that has never been known to be shown in your disease before might. And I really like the idea of, just seeing something in the lab that no one's ever done before.
So you're sifting around in uncharted territory.
Yeah, all the time. But I really like that. And actually a lot of some of that new stuff comes from working with people in other disciplines, like I mentioned, in the photonics fields, chemistry, and I've worked with a quantum physicist of all things as well. And it just means you go, okay, you've invented this nanodiamond and that can measure temperature. I want to see if that can show that wound healing's improved, or, whatever it is. And so you just get to try new things all the time. And for me, I really love that, and it's exciting, yeah.
But it might not be as sexy as research translation.
Yeah.
when we see the pointy end of the stick where stuff is actually out there and being used. We're talking about the background and the first steps.
Yeah.
It has never been described that way before. But why is it so important to have that foundation there?
Yeah, I mean, the thing is, like, we need to keep discovering new things. It's not like heart disease is solved. And as we mentioned before, we have reached a plateau. I think we've made a really nice advance lately, but it won't be the whole problem. So there's this thing which is this paper that you mentioned before. The title of it was "Don't Turn Off the Tap," and the reason I called it that was like, if you stop the flow of new ideas, we are not going to keep improving on how we treat people and how we keep reducing the risk of heart disease, as well as all the other diseases as well.
And so need funding for discovery as well as translation.
Always advocating for funding for discovery. It really can't be underestimated how important it is.
You're a relentless researcher. How do you think that links to your relentless rowing?
I think it actually is quite closely related, because both of them require quite a lot of discipline. It requires, well, one's late nights, one's early mornings, but you have a high tolerance at certain periods of time and a lot of pain. So I think there's actually quite a lot of rowing scientists, actually, because I think those two things fit quite well together. A lot of physics in rowing as well.
Does the physics come into your mind when you're rowing?
A little bit. I might be thinking, I don't know if the gearing on this is quite right, and you'll think about the pivot point on your oar anyway. There's a bunch of and resistance as well, and there are quite a few things that I might think about. I don't necessarily go, that's physics, but it is physics related that I'll be out there thinking, how can I improve things? Yeah.
And you took it really far as a younger woman, and you're still training and competing really hard now. Tell us a bit about your history with rowing.
Yeah. So I started rowing, when I was 14 years of age, and then we won schoolgirl nationals, and that really did within 10 months of me starting rowing, and that really did sort of immediately make me love rowing. And then after a couple of years, I made the junior team, and then the under-23 Australian teams in the women's fours. We won both of those, actually. And then I did try out for the Olympics for 96 or in 95, and I did not make it.
Robbed.
Robbed.
Clearly robbed after all that success.
Yeah, it was a real wake-up call, because I actually did have a really nice progression, but, just probably wasn't quite big enough, strong enough, or technically good enough overall just to quite make the cut. But
you must have been close.
I was pretty good. But I was pretty close. Got to sort of the final kind of selection regatta and then didn't make it.
That must have hurt.
It really did hurt.
Still hurts?
It's still better now, but that was in 95, 96. So I'm kind of gradually getting over it. But what it did cause me to do is literally throw myself into science. So I got home from the regatta where I didn't make it, and I got this letter from the University of Adelaide saying we would recommend for you to do an honours degree because of your grades. And I'm like, I think I'm going to do that. So I really threw myself into my honours year that year, and I think it kind of saved me. I kind of went about it like an Olympic athlete, worked really hard in that year, and I got a good mark, but really loved it as well. I really, really loved it. And after that, I just never really considered doing anything else.
But you've applied that discipline to your working life.
Yeah.
And still rowing now as well.
Yeah. So I still row now. I've returned to it as a masters athlete. Yeah, so I'm probably about 3 seasons back now, and, we train quite a lot now, but I love it. I think it's been a lot better for me to have rowing in my life again. And even though it can be quite hard to juggle with work, I think it makes me more efficient and a bit tougher, and probably don't have time to stress about things as much. So I'm in currently in the South Australian State Masters 8 that will compete at the Nationals at the beginning of May, so in a few weeks' time. So, yeah.
And when you're pulling, you're not thinking about science.
That's right, which is really good. So I was at the start line at the race, and I am not worrying about the grant or the paper that got rejected or even the grant submission time that might be coming up. So it is actually free your mind from work, and I do think that's healthy. And I think it's healthy for me, yeah.
Any other hobbies, or is it just pedal to the metal?
Yeah, I think I don't really. Even before I returned to rowing again, I probably would have said rowing was my hobby because it's kind of science and rowing, and I have an 8-year-old and a husband as well. So that really fills up the time, actually. So I don't really do anything else.
Do you ever go for a leisurely kayak or anything like that?
Oh, yeah, no, I have done. Oh, not for a while, but I have done a bit of kayaking. We did some multi-sport races when I had my time in Sydney because it's really beautiful to go kayaking around the harbour there. So I've done a little bit of kayaking, yeah, which is lovely, yeah. So I do a little bit of that, and I do like. I can't surf, but I like going to the beach, and I'm not too bad at boogie boarding. I don't know if that's too embarrassing to admit, but I actually really like doing that in the summertime. Yeah.
Okay, so there is a time to take a breath there at some point.
Oh yeah, definitely. Yeah.
One thing we haven't mentioned is your relationship with your dad, who was a scientist as well, and he really spurred you on to become a researcher yourself. Just tell us a bit about that.
Yeah. So my dad was a scientist. He's a very clever chap. In all science, to be honest, but he was a water chemist as his career, and actually was the director of SA Water until he retired. And then, like many people who retire, he then came back and actually was the chief scientist of South Australia about 10 years ago. So, yeah, and always supported my choice to be a scientist. In fact, really not pushed it on me, but really strongly encouraged to remain in science, not consider medicine. I don't know if he thought that would be better for me or if he had some strong bias, but it was, I guess, and also seeing his example of the amazing things that he was able to achieve in his science career as well also inspired me.
And he was able to teach you stuff at home?
Yeah. So, I mean, he was the best maths, physics and chemistry teacher someone could have hoped for. He was amazing. He remembered all of his year 12 science and into university as well because I did a science degree. So he was able to help me through all of that.
I'm guessing he didn't do it for you though.
No, he did not do it for me, but he was able to teach me anything I didn't know. So I was very lucky in that regard.
The homework was full on in the Bursill house.
Homework had to be done. That's right. And you had to be good at it, yeah. Yeah.
So where do you envision heart health research is going to be by the time you end your career?
Yeah, so I guess what I hope is that maybe we've solved it. So maybe the risk of having heart disease is close to zero. I believe that will be achievable. We will work out a way for early detection to maybe there's a blood test or something which can identify the earlier stages of disease, like I said, or anyone who might be susceptible so they can be screened or provided with treatments right from the beginning, because it can be reversible, it can be treated. It's the problem is when it gets too far, then it's hard to start doing things about it. So that's where I hope heart disease ends up, is that it actually is something that very few people have.
What about a drug that can just make the plaque vanish just like that?
Well, that would also be good as well. I mean, that you never know, right? You never know. There might be something like that. That sounds like a more physical kind of intervention where you could just scrape it out. And actually the surgeons do that. They do go in and they will scoop out plaque. But I mean, that's really invasive procedure. But if there was a miracle drug that could just make it vanish, that would be amazing. But once again, the trick would be to know, did that person even have it in the first place?
It's never that simple.
Yeah, that's right.
Thanks, Chris.
No worries. Thanks for having me. Yeah.
If you want to learn more about SAHMRI and the researchers working to build a brighter, healthier future for you and your family, head to samhri.org.au.